All right, we're live. Good morning, everyone out in TV land. Let's see. So, I had a couple of questions, or I've had a question on the homework. I'll probably do a little video on that just to cover it for everybody, rather than doing it right now. Yeah. Second, exams have been posted. It's easier for me to hand them out in lab, and that's what I'll do. So, or if you want to come upstairs and grab it, that's fine too. But there you go. Yeah. Any other thoughts? It's very quiet out in the audience today. We don't have a band. It's Friday. I don't know if that makes any difference to anybody. All right. It is so quiet. So, we've been talking about the organization of the plasma membrane. The fact that we have the lipid bilayer and we have the charged end facing the outside, the charged side facing the inside, because both the external environment and the cytoplasm are going to be ionic and polar and aqueous. We've talked a little bit about membrane biosynthesis. We have discussed the idea that we do have asymmetry in the leaflet, and I don't think anybody really knows exactly why that is or what purpose that serves, but it is there. We know that the phospholipids can diffuse this way, laterally, pretty easily. So the fluorescence experiments have shown that. Fusion experiments have shown that. We also can see that phospholipids can twirl, but what they don't do is flip-flop. That actually requires an enzyme known as flipase. And that is how the membrane is being biosynthesized, is that there's one side's being decorated and phospholipids being inserted, and then the whole section of it will go whoop and flip, and you'll work on the other side. Let's see. We know that we've got a whole range of proteins here in the membrane, and pretty much all the proteins are glycoproteins, and we'll be talking about protein glycosylation later, but just keep that in mind. It is a linkage to specific amino acids and serines, I think, regardless. We also know that this protein system, we have transmembrane proteins, we have peripheral proteins, we have proteins that are just on the surface on one side or the other. So integral membrane proteins, we remove those by using detergent. Peripheral proteins, we more likely can knock those off by using a salt wash. So now what we're going to start getting into is membrane transport, actually how we get stuff across the membrane. The fundamental point here is the fact that membranes serve as building compartments, and so we can accumulate stuff in a higher concentration on one side versus the other side. And this becomes a really important point. And, well, there are two really nice introductory figures that we will throw up and or throw out. Let's see if the computer will cooperate. Now, I like to draw, but there comes a point where I go, okay, this figure is nice. So, let's just look for a moment, because it's here. All right, so if we think about stuff that cells have to take up, and I were to ask you, tell me two things that cells have to take up, one thing might be, we're going to have to take salts up and back and forth. We've got to take water, it has to go back and forth. Glucose has to come across the cell membrane because the cell's got to eat. And these are things that carry a charge, and they may not be able to pass across the membrane very well. And so we have just a little diagram and a summary here of things that can and cannot pass. And it looks like this. All right, so first off, we have the difference between a protein-free artificial lipid bilayer, a liposome. And just as a little point, the RNA vaccines are being incorporated into liposomes. They don't have any membrane proteins. They pretty much have the concentrations inside versus outside of stuff. But with a cell membrane, we've got these transporters that can move things in, move things back and forth, as a channel, move things out. We have pumps, we have gated pumps, and so we've got all kinds of ways that we can adjust the concentration. But at least as importantly is this figure, which is, you don't got to memorize it, but you want to kind of keep in mind what's going on here. The idea that because the phospholipid bilayer has that lipid component, it's limiting what can pass across the membrane. And we're going to go through some terms here, like diffusion and passive active transport in just a moment, but for a moment here. Small nonpolar molecules, oxygen, carbon dioxide, nitrogen, steroid hormones. Keep that in mind when we start talking about hormone mechanisms. Steroid hormones and these other molecules, they can pretty much diffuse across the plasma membrane unimpeded. Now, we then have small, uncharged, but polar molecules, water, ethanol, glycerin, and mostly they don't pass the membrane, but they can a bit. So water is actually kind of an interesting one, and we'll talk about it later, but it may actually be able to kind of wiggle its way in between the cracks. There also is the idea of what are called aquaporins, which are channels that allow water to get in and out. But regardless, for sure we have larger uncharged molecules. Polar molecules like amino acids, glucose, the precursors of nucleotides, these barely can get across the plasma membrane. They pretty much are rejected. And then, of course, we've got stuff that have a distinct charge to them, all our various ions, and forget about it. They're not going across the lipid bilayer. So now, we have to have a way of taking these things up. And that's a nice figure, but we're going to get to it in a moment. So this brings us to actually a really cool experiment that books used to talk about. A guy named Overton did an experiment back in the 20s that was really quite definitive. Before we get too much further, though, let's come up with a couple of terms, though. Let's define these terms. The first term we need to define is diffusion. Now, diffusion is simply a thermodynamic process. Can you see that? D-I-F-F-U-S-I-O-N. Diffusion. This is a thermodynamic process. This just happens. And we can define diffusion as the movement of molecules from an area of higher concentration to an area of lower concentration. So the concentration, I decided to spell it out this time. This is a relative set of terms here. So you got higher versus lower. What we establish is a diffusion gradient. And if you don't have an area of higher concentration and an area of lower concentration, then you have achieved equilibrium. And at that point, there is no diffusion gradient. So my favorite example is just to sort of think about how diffusion works, because it is simply a thermodynamic process following along with the second law. That is, systems tend towards disorganization. If we have a bottle of Axe body spray up here, and all the Axe body spray molecules are stuck into one can, we have established a bit of organization. Now, if we close the room up entirely, turn the fans off, seal the room up completely with duct tape, and crack open that can of Axe body spray and put it up here, we have an area of very high concentration and then an area of low concentration. Fairly quickly, those molecules begin to move around, and within 5 or 10 minutes, in the back corner, you're going to go, smells like high school boy locker room. I don't know what. I used to use Old Spice aftershave, and then it would say, it smells like being pulled over by the police, but that's not funny anymore. So we'll brag on high school boys. The Axe body spray is moving about, and after a while, who knows, an hour, we end up with the same equivalent number of molecules in that back corner as we do in the can over here. At that point, things are still moving, but there is no gradient anymore. We've achieved equilibrium. And just to demonstrate the thermodynamic nature of this, you can imagine if the room is really, really cold, the diffusion is going to be slower. The movement of molecules is just slower. And so it'll take longer for equilibrium to be achieved. It's one of the ideas that, in Chicago, in the summertime, as you're walking around on the streets, there's all kinds of delightful smells. In the wintertime, the smells are significantly attenuated. And you might think it's because, it's colder weather, stuff isn't rotting. But the sewer is still probably the same temperature. The difference is the diffusion rate is so much less. So your trash will smell apparently less nasty because it doesn't diffuse as fast as it does on a warmer day. The idea here then, if we raise the temperature up, and you can imagine this, if we were to take the Axe body spray and heat it up, and heat the room up, very quickly it's going around and moving around that much faster. So the diffusion idea is strictly a thermodynamic principle. It is simply moving around from an area of higher concentration to an area of lower concentration because it is the second law of thermodynamics. So diffusion just happens. We don't have to worry about that. We establish a diffusion gradient. And now the whole point then behind membranes and cell operations is we can build diffusion gradients and take advantage of that energy, or we can use existing diffusion gradients and take advantage of that energy, and we can move things around and predict how things are going to move based on the diffusion gradient. So that's the driving principle behind all this biz, is either going with the gradient or going against the diffusion gradient, or allowing the diffusion gradient to use that energy to move things around. But what we have to now take into place is the fact that we have this phospholipid bilayer, and as that chart just showed, not everything can move across it. So diffusion is going to be limited because the phospholipid bilayer, depending on what we're trying to diffuse across. So here's Overton's experiment. It actually gives us a really cool insight into the way we can think about movements of molecules across the plasma membrane. So if you recall, we were talking about some experiments being done in which people were taking like red blood cells, calculating the surface volume, extracting them with chloroform to pull all the membrane components out, and then floating that on top of a little drop of water and measuring the amount of phospholipid that's present. And that's where they determined the fact that the volume of the cell is half of, or not the volume, the surface area of the cell is going to be half the surface area of the total lipid that you can lay out. And so that means, okay, it's easy to explain this as a bilayer. So the next thing was beginning to look at how molecules move across this bilayer. So this is what Overton did. He built a little apparatus in which he had, we can imagine, a little beaker, and there is a glass slide that divides the two halves. We have solutions of either half. You then drill a little hole right here. And what you can do is put in a glucose solution on one side and watch it diffuse. And so over time, the glucose is diffusing from one side to the other. All right, so this is a system that we can monitor, and glucose is real easy to assay for, even back in the 20s. So what he did was extract phospholipid, and he was, I think, using red blood cells, but you can imagine taking a little droplet of egg yolk and suspending this bit of membrane across this hole. And now watching diffusion. So what we've got here is, just for the heck of it, let's say we've got 1 molar glucose over here. And this is now pure water over here. So we've got a really good diffusion gradient, and you watch what happens. With the phospholipid suspended across this, there is very little diffusion occurring. There is not a lot of movement. And what you can do is, by adjusting the glucose concentration, the more glucose there is, the faster it's going to diffuse across, particularly if there's nothing. You can develop a rate equation like this. So if we look at the rate, i.e. the glucose movement per minute, and plot that against the glucose concentration, ooh, good mark. Plot that against the glucose concentration, we get a pretty simple rate equation. So, let's just go back here. If there's no membrane at all, just the hole, as you increase the glucose concentration on one side, the rate goes faster. And so what you see is, with no membrane at all, you see a pretty steady rate increase, no big deal. Okay. Now, when you add phospholipid, just pure phospholipid, all of a sudden it drops, because the glucose is not able to diffuse across this phospholipid bilayer very effectively. So this is just a plain hole. This is phospholipid. So then what Overton did was actually extract phospholipid not from like egg yolk, but actually from red blood cells. And then he suspends this. And what he sees is rather surprising. He sees that. Now, we've done a little bit of enzyme kinetics. We should look at that and go, I've seen a rate equation like that. That looks suspiciously like what we see as we add increasing amounts of substrate in an enzyme-catalyzed reaction. And you'd be right. What he has discovered is, in fact, the glucose passive transporter. And glucose is, as a sugar, it has a stereoisomer. You have either D-glucose, the right-hand stereoisomer, or L-glucose. And it turns out, as far as glucose is concerned, the naturally occurring stereoisomer is the D form. You can create levulose, or left-handed glucose. When he throws levulose into the system, he sees this. That is, whatever is happening here is stereoisomer specific. So this is L-glucose. L-glucose doesn't work the same at all. It's like having phospholipid bilayer there. What he has discovered is the glucose passive transporter, and he makes the following really critical observations. One, this glucose passive transporter, it is saturable. It has a Vmax. And this makes sense. We got this glucose transporter on the cell surface. There's only so many glucose molecules it can hold. And we'll actually see in a bit. That the glucose transporter is, in fact, touching and holding on to the glucose and popping it in. There are other kinds of systems, like there are ion channels, that simply open up, and they don't exhibit the same kind of saturable level of interaction. But for glucose, it definitely does. Another thing that he determined, not only is it saturable, it is phenomenally specific. specific to the exact stereoisomer. So Overton has discovered the glucose transporter. We now know that it is proteins that are responsible for this process. I mean, he did other experiments to demonstrate this. One of the main points now is this is aiding our understanding of how the membrane's put together. We don't have the candy coating. That sugar is part of the proteins. There are proteins that are integral membrane proteins acting as transporters. So this is a really good experiment. It is saturable. It is specific. So now, from here, we can begin to look at different kinds of systems involved in moving molecules back and forth across the cell membrane. And just as a quick little note, because I ain't going to write all this stuff up, and you certainly don't need to memorize it, but it's a good little thing to keep in mind of the relative concentrations of different ions in a cell. As we're about to see here, most all ions are not present at equilibrium. There is something going on here. And so, if we look at the inside of the cell, the intracellular concentration versus the extracellular concentration, let's eyeball here a few things. Sodium is maintained at a relatively low concentration. Can you see that? Oh, I'll give you some figures. You got the book. Sodium is around 5 to 15 millimolar inside, and outside it's about 145 millimolar. So we've got a sodium concentration, always more sodium on the outside. Potassium is the exact opposite. There's always a higher concentration of potassium on the inside compared to the outside. Magnesium and calcium, this is a kind of odd way of looking at it, but there are parts of the cell that have very high concentrations of magnesium and calcium particularly, but they're sequestered in the different reticula and different vesicles. The cytoplasm typically has a really low level of magnesium and 10 to the minus 4 millimolar, a vanishingly small amount of calcium. Calcium actually ends up being a pretty potent trigger for a variety of different kinds of kinases. We keep calcium really low inside our cells. Outside, it's a good bit higher. Hydrogen ions, this actually is a way we can look at our pH. We have 10 to the minus 7.2 molar, pH 7.2. And outside, pH 7.4, really fairly similar there. Chloride is, again, very low inside, very high outside. We will be paying particular attention to sodium and potassium. The other ones, just remember calcium. Don't be putting calcium in your cytoplasm unless you've got serious plans going on. All right, so now, we can define different kinds of protein systems. And these terms we've got then, we have a transporter in which we've got the actual binding of whatever it is we're passing the solute. And then we've got a channel. And this is just basically a pore. And so the Overton was looking at the glucose transporter. But let's go back to the board. And look up. What we're doing is defining a whole bunch of terms right now. So we can now begin to kind of build a table of how things are moving. And let us do this. So we have ions that are moving, or molecules that are moving with their diffusion gradient. And this is defined as passive transport. So passive transport is going to be using some kind of protein, either a transporter or a channel, the transporter being saturable and specific, and the channel being significantly less so. But the passive transport system we can be critically defining it as uses the existing diffusion gradient. Meaning we're going from high to low. So that's passive transport. And we can further then, under passive transport, break it apart into a transporter. And this is actually binding to the solute, or the material that we're passing through. Or we can have a channel. The transporter is significantly more specific and significantly more saturable. The channel is a bit more of just a plain old tube. All right, we have ways of regulating that hole, but still it's just a hole. Okay, so this is the way we think about the movement in the passive way. And actually, let's throw one more up, just to make sure we're being complete here. And do you have room on your screen for one more? No, you don't. We'll do it here. What we refer to as just simple diffusion. Simple diffusion is primarily the realm of water. Water is able to move back and forth just based on diffusion. So we can move things across with their gradient. All right. Now, as it turns out, if we're going to begin accumulating stuff, and looking at that chart, clearly we do, we've got a lot more potassium inside than we do outside. There is another class of transport system, and it is this. We have active transport. And active transport we can define in the following way. This is now a movement of molecules against the diffusion gradient. So against the diffusion gradient, meaning that we're accumulating stuff, or we're pushing it out. We're going from an area of lower concentration to an area of higher concentration. This is violating the second law of thermodynamics, and we know if you break the law, you've got to pay. Consequently, this is going to be energy requiring reactions. And we can further subdivide active transport into two forms. We've got what's called direct active transport. Direct active transport is actually using the phosphate from one of those ATPs and changing its shape to generate the force and the change in protein shape to actually drive this movement of ions. So direct is directly using ATP as the energy source. So that phosphate is causing the protein shape to change. We also can have indirect active transport. Now, indirect is going to use an existing concentration gradient and using that to drive another uptake against the concentration. So with indirect, we're going to use an existing gradient to drive the active uptake. let's see. So to drive, how about that? To drive movement against the gradient. Can you see that? Sure, you can see that. All right, so this is a good thing, and let's be honest, this is like one of the 15-point test questions. To define passive versus active and break it down and basically throw back this chart back up at me. Passive transport using the existing diffusion gradient, movement from high to low. We got transporters, we got channels, we have simple diffusion. Active transport, movement of molecules against their diffusion gradient, meaning we're going from low to high, we're accumulating stuff, going to be requiring energy, and in a sense, this is how we can determine or how we break down the two categories. If it is directly using ATP, it is direct active transport. If it's using an existing gradient, then it's indirect active transport. So we now are going to go through a couple of the different transporter systems and see how they work. So, one other definition that is worth throwing out and throwing up. You may have had this defined to you before, but maybe not, and that is osmosis. Osmosis is a specialized kind of diffusion, and we'll be talking about it a lot, so it's important to define it. Let's define osmosis in the following way. Good mark. Osmosis is diffusion of water across a semi-permeable membrane. So let's take this apart here. The diffusion of water, meaning water is moving from an area of higher concentration to an area of lower concentration, and it's moving across a membrane that is semi-permeable, specifically not to water, but to different ions. And this actually, is a good exemplification of what happens as we start moving ions around. Pause for a moment. So osmosis is a specialized case of diffusion, and this, we call it a couple of terms here. First off, you might say concentration of water. How do we have different concentrations of water? How? I don't understand. Water's water. But think about this. If you were to take a measuring cup and fill it to 2 cups, and now you're going to take a cup of sugar and dump it in there and start stirring it up, what do you think is going to happen to the water volume? It's going to go up. It's going to go up. Okay, really? You got a beaker, you got a cup, you dump it in there, the volume goes up, it splashes on the table. Oh shit. Stir it up, it goes into solution, it still don't go down all the way. So the addition of this stuff, which, by the way, we refer to this stuff as solute. Solute equals dissolved stuff. If you can dissolve it, it's a solute. Solute displaces water. And so the way to think about this now, like let's get some graduated cylinders out here, or your measuring cup. You're going to take a cup of water and set it here. Now you're going to take a cup of water and add half a cup of sugar to it, stir it up. The volume goes up. When you take a half cup of that, you actually have some water left over. You got 2 half cups. One actually contains, pure water, more water molecules than the water with the dissolved solute in it. So the act of dissolving something into water, it displaces some of the water molecules. You can think of it in a lot of different ways. You have less concentration of water. So now, because of this, we actually can think about a water gradient. And we can set up some interesting things that will happen with an osmotic gradient. Specifically, let's just look at this idea. If we take a beaker of pure water, and this could be tap water, really, but we got pure water. And now we put in a red blood cell. So here is our red blood cell. Kerploop. The concentration of water out here is straight high concentration of water. Inside the cell, there is in fact a lower concentration of water because we have all these dissolved salts, we have all these dissolved proteins, we got a lot of solute in here. And so, from a physiological perspective, we've got about 0.9% salt. That is physiological saline. And because there's salt here, it has displaced some of the water. The water concentration is less in the cell than it is out here. The diffusion gradient is set up. Water is going to move from an area of higher concentration to an area of lower concentration, i.e., water begins to move in, and as it does so, our cell goes from being happy to being a little bit upset to being, oh no, and eventually poop, and his guts float out. So the cell is going to start taking water up until it ruptures. If I didn't have a mask on, I'd go, but I do. It will pop. And we have set up an osmotic gradient. One way to always keep in mind what direction water moves is this sort of general mnemonic or whatever, and that is water follows salt. Because in cellular systems, you can't pump water. What you pump is salt. If you need to move water from one cell to another, you don't pump water. You start shooting salt from this cell either out or into the cell. Water moves towards the salt. That's how it works. Your kidneys do exactly that. If you need to pee less, you start pumping salt out into the surrounding tissues in the kidney. As the P to B is passing through the kidney, water is being pulled out through osmosis, and you can concentrate your urine quite effectively. Try it. You can also have less concentrated urine where you're not using osmosis as much, and you can pee out more. So this is how we can think about the movement of water. You may or may not have heard the terms. We have these relative terms. We can have something that is hypertonic, meaning it's got relatively high salt compared to whatever else you're talking about. You can have stuff that is hypotonic, meaning that it is low, like a hypodermic needle. Or you can have something that is isotonic, meaning that it's the same salt concentration on both sides. So just to set this up in a different way, at the moment, the cell is hypertonic to the water, or another way of saying this is we've placed this cell into a hypotonic solution. It's not enough salt. But what if we, instead of using pure water, let's go ahead and add some salt to this. So let's make this 5% salt, which is pretty salty, but you know. And now we have little Reggie, the red blood cell, at 0.9%. And water follows salt, so the water moves out and our little cell begins to shrivel up and become a raisin. The cell is now hypotonic, and the solution is hypertonic. He won't rupture, but it may not be very pretty. The cell shrinks, the water concentration drops, the proteins probably start to denature a bit. You may be able to revive this guy from that situation, but you may not be able to. So adding salt to stuff pulls water out. The only reason we need to go through this is when we start talking about the movements of these molecules, we can affect the osmotic gradient, and that's a critical thing. All right. So the first transporter we're going to talk about in the active transport side is a classically studied transporter, and you'll see why it's critical to salt. It is known as the sodium-potassium ATPase. The sodium-potassium ATPase. It may have been actually first identified as a system that breaks down ATP, consequently the ATPase. And then subsequently they realized, oh, it's also pumping sodium out and potassium in. And let me just see where we're looking at here. Figure 12-11 is looking at the sodium pump. Regardless, what we got here with the sodium potassium ATPase is you have 3 sodiums moving out and 2 potassiums moving in to the cell. So the sodium potassium ATPase is stuck in the membrane, pumping these ions back and forth, and the ATP is being hydrolyzed, and I think in this case it is in fact a direct kinase, but I'd have to look at that on Wikipedia. The point is ATP is hydrolyzing in this protein, kind of like the proteins in the cell membrane here, and you've got this protein complex. ATP is dropping a phosphate off, being converted to ADP, and that is changing the shape and the binding affinities of this protein such that we have the sodiums moving out and the potassiums moving in. One of the things that is known with this, and it actually ties into discoveries made later, is that you can drive the sodium potassium ATPase backwards, kind of synthetically, by putting it into a very high salt concentration on the outside. You can begin driving it backwards and actually make ATP. You can use it as an ATP generating system given the appropriate salt concentrations. But regardless, this is an example, a perfect example, of a phosphate changing a protein shape, and it's pumping sodium out. Now, the critical thing is, right now, your sodium potassium ATPase is pretty much in every single cell of your body cranking away and dumping sodium out, bringing potassium in, maintaining that critical sodium gradient. It is said that you use around 30% of your calories doing nothing but that. So even though you're not doing anything, you're sitting on the couch chilling, you are burning ATP, quote, burning calories, and one of the main ways you do it, a full third, is just pumping sodium out. That's how critical this is. Every cell, all the time, pumping sodium out. And that's because we're going to use sodium as a co-ion for bringing things in. We can depend always that the sodium concentration inside the cell is always low. We always have a sodium concentration gradient such that you can depend on the cell having low sodium. And so as long as the sodium concentration is higher in the external environment, we have a concentration gradient that we can use as a driving force. We can have a co-transport ion in the form of our friend sodium. That is the indirect active transport key. We have always sodium low inside the cell, higher outside the cell, and so when we need an existing gradient, we just turn to sodium and you're off the races. We use sodium co-transport to bring in just about everything. So you bring in glucose against its gradient using a sodium co-transporter. You bring in amino acids. You bring in anything you got to bring in against its gradient. We use a sodium gradient almost exclusively as the existing gradient. So the sodium-potassium ATPase, that's our direct active transport guy. And its job is to always make sure the cell has a low sodium concentration inside. It doesn't really care what you got to do with that sodium. It just says, yep, pump it out. Always pumping it out. And you think about, the fact that 30% of your ATP, 30% of your calories goes to just this. That's how critical a global kind of phenomenon this is. And in our last moment, there is an inhibitor of sodium-potassium ATPases, and that consequently a pretty nasty poison, a compound called wabane. And I like kind of saying wabane. Wabane, as an inhibitor of the sodium-potassium ATPase, will screw cells up in a big hurry. And one of the things that you can see is you throw wabane into like tissue culture cells, and it blocks the sodium-potassium transporter immediately. And over time, it takes about 30 minutes, you start to see the cell swelling up until they rupture. Now what's happening is the sodium gradient exists at the beginning of your experiment. You shoot wabane in, you can't pump sodium back out, but that sodium gradient is still there. Things are moving in with that sodium gradient, but now the sodium concentration starts to increase inside the cell, and we end up with the cell becoming hypertonic to its existing environment. Water follows salt, water moves in, and zing-zow, the cell pops open. So blocking your sodium-potassium ATPase, man, that is a great way to poison yourself. A metabolic poison that will get every single cell in your body. Don't eat it. And I'm thinking, I don't know where wabane comes from. I bet it's a fungus, but I bet it would kill you really bad. All right, so that's a lot of good concepts and things here that we've covered. And what we're going to pick up on Monday is how we can begin coordinating these things into different systems, taking glucose up in the intestines, and then actually looking at how the nervous system is using ion gradients to transmit nervous system signals. It'll be a good time. Join me, won't you? All right, so I'm going to probably put together a little song and dance about the homework to sort of help. And then we'll be passing exams out in lab just because it's easier for me to grab them. I'm right across the hall. And that's all I know. Ciao. Bye-bye.